Good evening, everyone, and thank you for coming tonight to our presentation. Tonight, I want to introduce to you Dr. Lonnie Nguyen, and she will be sharing a presentation on Eucharistic miracles. Dr. Lonnie is a professor of biological sciences at Mount San Antonio College in California. Dr. Lonnie has taught classes in the Diaconate Formation Program for the Diocese of Orange. As an expression of her belief in the true presence of Jesus in the Eucharist, she's developed this presentation to share the scientific evidence that supports Eucharistic miracles. This presentation is based on the book A Cardiologist Examines Jesus by Franco Serafini, and I noticed you can find it online at Amazon or other places if you would like to get a copy of that. Let us please welcome now Dr. Lonnie.
Okay, can you hear me? Yes. Good, perfect. All right, well, thank you for being here. I know it's a Monday night and you've all worked probably a full day, so I really appreciate your presence. And if you need to, stand up and stretch and get the blood circulating, please do so. I won't feel bad. Like I said, I'm so appreciative that you're here. I also want to start off with saying thank you to Father Ed and the staff for organizing all this. Father was here last night with us downstairs, and so he already heard this part. And for those of you who are here a second time, tonight's talk is slightly different. It's less interactive because it's a lot of you, and I'm trying to keep this to an hour. So I'll do more of the talking, but hopefully, there will be some time for Q&A at the end. And I'm sorry, should I speak louder? Yes. Oh, I should speak louder? Yes. Oh, okay, all right. Wow, all right. Please let me know when my voice starts dropping. Thank you so much, okay. So anyway, I just want to refer to this book by Dr. Franco Serafini. It's A Cardiologist Examines Jesus. My husband and I just read this last summer, and we were so amazed. It's great information, and it's something that we've never read anything like this before. So as a teacher, I said, I gotta share with the world. I can, if I can, I would stand on the rooftop and shout it out to anyone who's willing to listen, because it's so powerful, and it definitely has impacted our faith, and I hope to share the same with you tonight, okay? So who are we? We are a deacon couple. Deacon Peter, my husband, my other half, and he was ordained in 2013 in the Diocese of Orange. That's in Southern California. He's also a retired IT geek, so all my technology I refer to him to help me. As for myself, I am a physician by training, but I am teaching. As you heard, I teach at a community college down in Southern California. And so I use that skill to communicate and hopefully to make things understandable and something that will stay with you after tonight as well, okay? We started our ministry, the website is missionroad.org, because that's what we're on, right? We're all on the journey, we're all on this mission, and the goal is to serve God, to share the good news, and to have others join us on the road. So that's the name, and if you want to know more about what we do, feel free to go to our website, okay? All right, so why are we here? Why this talk? This picture pretty much says it. I don't know if you realize, but a Pew Research came out not too long ago, and it surveyed Catholics as to, do you believe in the true presence? That is, do you believe God, Jesus, is in the Eucharist, okay? And raise your hand, have you heard of this survey? Oh, okay, well, quite a number of you. All right, so anyone who has their hands up, someone is close here. So do you remember the outcome? One-third of Catholics that was surveyed said that yes, they believe in the true presence. The other 2/3 said it's a symbolism, which is completely wrong, correct? And so with that, Bishop Robert Barron started the Eucharistic revival. And as you know, we're in that 3-year period right now. And so this talk, just for me, it just came perfectly. Now, I have to admit, a little confession, it's hard to understand transubstantiation, right? It's hard to understand that, yes, Jesus is in the consecrated communion host. Because honestly, it still looks the same, right? And the wine still tastes like wine, but we're taught by faith to believe. And so I struggle with that personally. And then this book came, and I'm like, oh, it's like my eyes were open. Okay, so as a scientist, I love science, and anything that's logical and makes sense to me, I try to pass on to you. And that's exactly what this book is all about. So we will start with the miracles. Okay, now I'm going to give you the Cliff Note version because it's a lot of interesting story, and I really want you to read it for yourself. It's rich in details. It's easy reading the first part, and so I'm going to do what Dr. Serafini did, in that I'm going to start with each one of the miracles. So the first one in the book is Lanciano, Italy. This is famous, right? Most people have heard of this. The miracle started somewhere between 700 to 750 AD, and a priest did not believe in the true presence. So that priest prayed on it and asked God for help. So one day, the priest was celebrating Mass, and it happened. The miracle happened right there during Mass. So right now, if you go to Lanciano, Italy, this is what you would see up in a monstrance, up high. That would be the body of Christ. That is actually heart tissue, okay? And then down below in that glass container are 5 pellets of blood, okay? Of course, it has been 1,300 years, so it's not what it was before. It has dried out considerably. It was there was no fixative, no chemical added to preserve it. It was just kept in air, okay? Now, in 1970, Dr. Lano in the area was commissioned to examine Jesus, okay? And my hat's off to him, because something that happened 1,300 years ago, Dr. Serafini wrote in his book, for Dr. Lano, the blood, the Christ, was like hard, like wood. But somehow he managed to take a tiny little bit of Jesus and run his tests, and you will see some of the results in a short while. Okay, so remember, that's the first miracle. The second miracle that's covered in the book is this one here, and that occurred in Buenos Aires, Argentina. Now, for whatever reason, Jesus decided to show up 5 times, okay? So the first appearance was in 1992, okay? And that was through a consecrated host that was abandoned. No one knows why it's there. It was soiled, it was dirty, so it could not be consumed. So the protocol is we take Jesus and we put Jesus in a cup of water, put it in the tabernacle, and then in about a week or two, with time, Jesus will dissolve. And once Jesus is dissolved, then that cup the content will then be buried in the ground of the church, the sacred ground, and that should be the end. Well, that didn't happen. So they followed the protocol. Jesus was in the cup in water in the tabernacle. A week later, went to check on it, and there's this red appearance that was miraculous. No one really understood what was going on. No one knows why, what's going on. But that was the first miracle. Now, after that, 10 days later, okay, May 1st was the first one, May 10th is the second one. May 10th, 7 p.m. Mass. The cup that held Jesus, there was blood streak on the edges, okay? During Mass, same thing, no explanation, no one knows. So that's miracle number 2. That same night, in the second evening Mass at 8:15, a different cup also had blood streaking down. So that's miracle number 3. And as if that wasn't enough, 1994, July, Jesus appeared again. Same situation. In a different cup, we have Jesus with blood streak coming down. Okay. And then lastly, in 1996, in August, another consecrated host was found. No one really knows how it got there or what happened. And so same protocol, they've done this already, so they know. They put Jesus in water in a tabernacle, and later on, a week later, they checked. Jesus did not dissolve. Jesus became red. Okay, so I tell you this, and no one really no one knows why. 5 different miracles occur in Buenos Aires in a very short amount of time. So the authorities, the bishop of the area, the first time in 1992, they kept Jesus in holy water, and then after a month, whatever reason, we don't know, they transferred Jesus into distilled water and capped it off. Kept Jesus in distilled water for 3 years before our current bishop, our current Pope, Pope Francis, before he became Pope, he was Archbishop of Argentina, right? So he was the one who authorized the scientific analysis to be done, okay? And you look at you find out the findings in a little while. But I just want to emphasize the miracles in that Jesus was kept in distilled water. Now, as I mentioned, a little bit of biology, okay? For a biologist, I was like, oh my goodness, why did they do that? And the reason being, there is a car headlight left in the parking lot, a Toyota Camry, license plate CFG6120. I don't want your battery to die on you, so if you have a Toyota Camry, your lights are on. Okay, it's gone. It's gone? Oh, okay, all right, thank you. All right. Okay. Back to the significance of keeping Jesus in distilled water. Okay, so this is a biology picture here. Cells, okay, animal cells. We have the cell membrane, and that allows for water to move in and out. If we put the cell in a solution that is of higher concentration than the cell, inside the cell, the water inside the cell will leave and the cell will shrink. If we put the cell in a solution of the same concentration, then there's really no change because there's going to be water moving in and out. But if we put cells in a solution that is lower concentration, as in distilled water, it's very low concentration, what happens over time is water will move into the cell, boom, and over time the cell will burst, which means destroyed, right? So when we read that they kept Jesus in distilled water for 3 years, that's a miracle that it did not destroy the cells that was there, okay? So let's move on to the third site, and this is Tixla, Mexico, 2006, okay, times fast forward. This was a retreat offered to the local parishioners. 600 people attended. And at the end of this retreat, the last Mass, the closing Mass, we have 2 priests and 2 religious sisters distributing communion. Okay? So one of the sisters, she's had this line, right, of people, and she's giving communion, she's passing out Jesus, and she looked down, and that's what she saw. Okay? There was something red on the consecrated host. So at that point, she halted the communion line, and she went and showed to the priest. This priest actually has already witnessed different miracles elsewhere, and he knew exactly this is a miracle. So he held Jesus up. 600 retreatants saw and witnessed the miracle, and thus this picture was taken. Okay, so wow, that must have been one heck of a retreat, don't you think? Now, for this, the first analysis was also in 2009. So 3 years. The bishop in the area decided to wait 3 years before starting to study Jesus. Okay, this next one, miracle location number 4, Sokołka, Poland. Okay, and this happened in 2008. It was a Latin Mass because they talk about the parishioner coming to receive communion, kneeling at the communion rail. And as she knelt, she saw in the corner a consecrated host that has been soiled. So she told the priest, and same protocol, put Jesus in a cup of water, put Jesus in the tabernacle, and instead of dissolving, as expected, Jesus starts to turn red. So this picture is amazing because if you see in the top corner, you still see the wheat from the communion host, right? But then you see this red, almost like blood clots, right? And that is intertwined. Okay? Underneath the microscope, the tissue is intertwined with the wheat grains. It wasn't like we have the wheat on one side and then you have next door the blood clot. So that is a miracle in itself. Okay, so after about a week or 2, they decided to take Jesus out of the water and put Jesus on the corporal. And from that point on, they kept Jesus in the monstrance in air. Okay, no fixative, no chemicals. And the analysis was done about a few months later in 2009. And then the 5th miracles mentioned in the book is this one here, Lano, Poland. And this is special. Christmas Mass. First thing in the morning, parishioners come to Christmas Mass, and the miracle, they found another communion host, consecrated. Jesus was on the ground, and the priest picked Jesus up and put Jesus in water in the tabernacle, assuming over time Jesus would dissolve. What? It didn't happen that way. And so Jesus turned red, and now they have that miracles also in a monstrance, and just kept in air, okay? No fixative, no chemicals. Now, because it is December 25th when it first happened, it was about a few months after that the bishop decided to give the scientists a chance to examine Jesus, okay? All right. Now, so in the book, Dr. Franco Serafini talks, gives a really good overview of what happened at each location. It's excellent reading, fun reading, so I highly recommend you at least read those first 5 chapters. But in addition, he also brings in the Passion Cloth, okay? What did the scientists find when they examined the Passion Cloth? So I'm sure you all know what this one is, right? Okay, I see a lot of nods. Okay, so this is the Shroud of Turin. Lots of controversies, and Dr. Serafini does an excellent job of going through the controversial stories that comes about. But honestly, this is another miracle that has been extensively studied by scientists. And I'm going to show you later on the findings of the cloth. I just want to let you know that this is also brought into the book. Now, two other passion cloths that are not as well known as the shroud is this one here. Passion cloth number 2 is the Sudarium of Oviedo. Raise your hand if you have heard this. Oh, wow, excellent. Okay, you're better than me because prior to reading this book, I have never heard of this. I'm like, what? Okay, so this is believed to be the cloth that was wrapped around Jesus' head during burial, okay? And it's not very big, it's about yay big, okay? Now, how do we know? Well, we don't know for sure, but scientists examined it, and they looked at the stain on the remaining cloth, right? So I don't want to get too gory, but I've got to tell you the scientific detail. The stain on the cloth correspond with bodily fluid that came out of the person's mouth and came out of the person's nose after death, and also there were blood clots that stained the cloth that correspond to something that would have been consistent with the crown of thorns, okay? 6 out of 8 points on this cloth correspond to the same points on the shroud. So you're like, well, okay, I would need 100%. No, I mean, 6 out of 8, come on. That's a pretty high match, right? So highly likely that the person that the shroud wrapped around is also the same person that had the cloth, the Sudarium, okay? So that's also been studied, and we will look at the scientific results from that as well. And then the third passion cloth that's mentioned by Dr. Serafini is this one here, the tunic of Argentile. Now, raise your hand if you heard of this. Oh, okay, well, it's great. So for all of you, you're just like me. This is brand new, right? So the tunic is seamless, is made of wool, and it's believed to be what Jesus wore on his way to Calvary, okay? So if you can imagine, we know about the passion. We know Jesus was beaten, he was bleeding, he was, as he carried the cross. So the bleeding on the tunic shows that Jesus carried a cross, not just a horizontal beam, but he actually had the full cross on him as he went up. And I just got to throw in there, remember the cloth that's wrapped around the Sudarium, right? The cloth around his head. They found bleeding where the nose would be, okay? And with the bleeding, they found soil. They found vegetation that would be in the area that would put that person in the place of Jerusalem about the same time period, okay? So what does that mean? It means that this person fell face down, like bam, he hit the ground and he couldn't use his hand to brace himself, okay? It's because he had the cross on him. And so when I read that detail, it just brought me to tears. I can imagine the pain as you're going down and there's nothing you can do, and it records that the tunic records the cross, and literally, the compression that as he fell onto the ground. And we know Jesus fell not once, not twice, but 3 times, right? So, yeah, it's an amazing story. If you don't know about this, Dr. Serafini does a great job explaining the history of this, which is another, it's just mind-blowing. But I can't tell you the story right now because I got to move on. So in a nutshell, I just want to show you the overview of these 5 miracles, okay? When it happened, and when the scientific analysis were done. As you can see multiple times, we don't, as scientists, we're a little skeptical. We don't do things just once. We want it to be repeated, and we want to make sure that the results are consistent, okay? So talking about science, I like to acknowledge three big names who has contributed greatly to this entire Eucharistic miracles examination. So first and foremost, Dr. Franco Serafini, he's the one who wrote the book. He's an Italian cardiologist, dedicated a few years of his life to look at the scientific evidence, okay? He himself did not do any of the research in terms of examining Jesus himself, but he went to the location, he interviewed people, he talked to the clergy, he read all the scientific reports that were generated, and he took all that and put it into the book. So I'm so grateful for his work because he made things a whole lot easier for me and for all of us who are going to read his book. He's an excellent writer, by the way, very clear, very easy to follow, has a lot of footnotes, so if you're not into the science, you can look at his footnotes and get an explanation of what he's talking about. Okay. The middle man, Dr. Ricardo Castellón Gomez, I gotta say, he started off as an atheist. Okay? Our current Pope Francis, back in 1995, commissioned him to lead the research on the Buenos Aires miracle that happened in 1992. So in 1995, Dr. Gomez came on board after 3 years of Jesus sitting in distilled water. And, you know, he was chosen, first of all, because he's famous, big name. Number two, he's an atheist. Okay? As scientists, we do not want a bunch of Catholic scientists say, oh, okay, of course it's a miracle. Okay? So we want an objective person with no agenda. And then last but not least, the third scientist is Dr. Frederick Zugibe. He's a physician, he is a pathologist who has himself have done over 10,000 autopsies. That's a lot. And his area of expertise is the heart. So you're going to see how he plays a critical role in helping to determine what exactly is going on. Okay, so those three are the biggies. There are many more scientists who are, involved, and I just don't have the time or the slides to acknowledge them all. But I just want you to understand that, especially with the later miracles, the one in Poland for sure, those in Poland and Tixla, Mexico, they had teams of scientists because, as time goes on, the technology is much better, the scientific analysis is much better, and they didn't send Jesus to just one lab. They sent Jesus to multiple labs. They got multiple experts to consult. Okay, so many teams of scientists all over the world shared in this work. So that's typical of science. Science is we want to be open-minded. We want to look at the data, and then we look for the patterns, the consistency in the data. If it's not consistent, then we tend to throw it out and say, it's not the same. Okay, so please keep that in mind. Scientists, we ask a lot of questions, and we debate. So it's none of this, don't ask any questions. It's none of that, okay? So let me kind of go back to Dr. Gomez. So he was involved in analyzing, spearheading the miracles that happened in Argentina, right, 1992. Well, he was just so amazed that 1990, actually, fast forward, Tixla, Mexico, right? Dr. Gomez, South American man, Mexico, not too far away. He was there. And after 3 years, he said, I like to lead this. And sure enough, he got that request. And so he also led the scientific analyses on the Tixla, Mexico miracles. Okay. Long story short, he's now a Catholic because he cannot explain scientifically, and therefore it has to be supernatural. Therefore, it has to be divine. And to his credit, he started a nonprofit that funds the research because these are expensive processes, right? Getting researchers all over the world to examine Jesus without telling them the source is incredibly difficult and expensive. So a lot of time when they send Jesus to these various laboratories, they did not disclose where the sample came from. They just said, would you please run, analysis on this and tell us what you find. Only afterward, then they disclose, oh, this is blah, 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 whatever. So we do that in the scientific community because we don't want the scientists to be biased, right? They say what they say. They can't say, I'm going to hold back because I know where this comes from, and this doesn't make any sense. Okay. So that is all going on in this. So let's jump right into the scientific analyses. First and foremost, quick biology. This is an animal cell, and in the animal cell we have the nucleus, and in the nucleus you have DNA. That's our genetic material, right? So all human cells have this makeup. So of course, and this is what DNA looks like. They're double-stranded. Okay, so if you look at each level of the double strand, we call that a base pair. Humans, we have 3 trillion base pairs in our DNA. Okay, that's a lot. There's a lot of zeros there. And so now, we have sophisticated technology where we can analyze DNA. Okay, so DNA analysis we conducted on the Eucharist, and they confirm it's definitely human DNA. Not once, but multiple times by multiple labs, okay? But interestingly, they could not profile the DNA. So let me kind of explain what DNA profile is. Actually, it's synonymous with DNA fingerprinting. Okay? So raise your hand if you're familiar with DNA fingerprinting. Oh, okay, just a few. All right. So let me kind of digress and explain DNA fingerprinting. Kind of think of it like our fingerprint. Okay, we can all relate to that, right? It's unique to us, agree? Okay, so the pattern, the world on here tells us is us, right? I mean, it doesn't say, oh, this is Lonnie Wynn. No, it just say, I had this specific pattern, and so if I was to touch something and I leave my prints behind, they can match that with this and say, oh, you touched that. Does that make sense? Okay, so with DNA, it's similar, okay? So instead of the world pattern, is that ATGC that I showed you last time. Okay, so that ATGC, that's your sequence. And that's just 4 letters, ATGC, and it gets repeated 3 trillion times, okay? So that, in a sense, is your DNA fingerprint. Now, it takes too much money to look at the whole entire 3 trillion base pairs, so we cut short. In other words, instead of looking at the whole palm, you zoom in on the finger pads, right? There's no point looking at the whole thing. You could, but that's very expensive. Same thing with DNA fingerprinting. They zoom in certain sequence, certain area, and it's going to be unique to you. Okay, whatever the sequence is in that specific area is going to be unique to you. And currently, in application with like CSI, crime scenes, investigation, if they look at 15 sites, and that 15 sites match your DNA fingerprint, that's pretty much you. It's 1 in a billion chance that you're going to have someone not related to you to have your exact same sequence in those 15 sites. So it's incredibly accurate, okay? And it's currently used by crime labs all over the world. So with the miracles, that's what they try to do. They try to fingerprint Jesus. What do you think? Not possible, okay? So keep that in mind. Try as they could, they try hard, they did it multiple times, and it never came out. We were not able to fingerprint any of the miracles. You and me, easy. Okay? They can easily fingerprint us, not a problem. So for many people, it was disappointing, but for Dr. Gomez, he said, aha, I know this is supernatural. Because if we were able to fingerprint Jesus, that's way too easy, right? And he would probably thought it's a fake. But this actually made Dr. Gomez one of the reasons why he believed. Now, let me compare to the consistencies in the Passion Cloth. Passion Cloth, remember those three, the shroud, the tunic, and the sudarium? They ran DNA analysis also on those specimens, and they come up, by looking at the genes, looking at the alleles, they know that it belongs to a Jewish male, okay, who is healthy, because they look at one specific disease that is known to exist in that time, in that area, in that population. And this Jewish male did not have that disease, so therefore he was a healthy person, okay? And that pretty much is in line with what we know about Jesus in the Bible, right? He was a healthy Jewish male. Okay, let's move on to blood, okay? So lots of blood in the Eucharist, miracles, lots of blood in the shroud and the sudarium and the tunic. And raise your hand, you remember blood type in high school. Oh, okay, all right. Raise your hand, you know your blood type right now. Oh, okay, that's better. All right, so blood type, many of you probably already heard, Jesus is AB blood, right? Okay, so let me kind of delve into the significance of that. Just curious, I'm trying to be interactive so you don't fall asleep on me, okay? If this is gradient here, the gradient, if you hold your hand very high, that means that, so this is 0, okay, 0% of the population, 50% of the population, 100% of the population, okay? So AB blood, what do you think? 0%, 50%, 100% of the population. So where do you think is AB blood? Oh, yeah, I see a lot of hands up. Okay, you are correct. Depending on the population, depending on the geography, anywhere, on the average, globally, okay, internationally, 1 to 5% of the population has AB blood. 1 to 5%. So yeah, it's very, very small. Okay. Now, one lab in Bolivia was able to also run the test of positive or negative blood on Jesus, and this scientist, she claimed, she said, definitely Jesus was negative, AB negative blood. So what do you think? Is negative blood rare or common? Okay, wow, you're good. Okay. So, extremely rare. Majority of us have positive blood. Only about 15% of the population has negative blood. Okay. So if you do the quick math, okay, probability of having AB negative blood is, let's say, 5%, multiplied by 15% for the negative. That's 0.75%. In other words, 1 out of 133 people would have AB negative blood. That's very hard to find, correct? Okay, but these 5 miracles, when they ran the test on the shroud, on the sudarium, on the tunic, guess what? Time and time again, AB blood. And so, yeah, it's AB blood. There's really no contesting that. And so you think about people who say, oh, this is a forgery, this is a fake. Oh my goodness. Lanciano, Italy, 700 AD, we didn't even know about blood type, right? Blood type was only discovered in the 1930s, okay? The person who found that got a Nobel Prize in medicine for that. So we didn't know about blood type. And so for every test over the time, over these, different specimens to come out with AB blood, that's pretty a strong definite Jesus is AB blood. Okay. All right. And in addition to running the blood type, they also ran what they call electrolyte panels. Electrolytes are your calcium, your potassium, your magnesium, your chlorine. You know, we all do that whenever you go get your blood drawn. Your physician will order a Chem 7, panel 7, and then they'll say, oh, well, your potassium's kind of low, or your sodium's kind of high, or, you know, something like that. And that's usually a strong indication of how healthy you are. If you have, say, kidney problems or some serious stuff going on, those numbers will be abnormal, and that would tell you that the person has something going on. Okay. But in Jesus' case, always normal. So that also confirms that Jesus was healthy. Okay. All right. Now, another interesting finding. Okay. So here I show you a picture of red cells and white cells. So those cells that are not red, those are under the category of white blood cells. Okay. So Jesus, in the Eucharist that was analyzed, multiple labs said, oh, interesting, there's a really high count of lymphocytes. Lymphocyte is a specific type of white blood cells, and so the condition is called lymphocytosis. And in addition, he had a low amount of a specific antibody, a specific protein. So fancy word is hypogammaglobulinemia. Now, many conditions might result in lymphocytosis, and certain other conditions will result in hypogammaglobulinemia. However, there is one condition where both would exist simultaneously, and that is someone who is enduring tremendous physical, emotional, spiritual stress. Major trauma will trigger both conditions to exist simultaneously. Okay? And that two conditions existing simultaneously is transient, meaning it won't last forever. It will last for a day or two, and then it will disappear. The fact that the blood, the Eucharist, saw this, it means right then and during the Passion, that's the blood sample at the Passion. Not when Jesus was healthy, not when Jesus was a little boy, at the Passion. Okay, does that make sense? Okay, so keep that in mind. Because now we're going to go to another examination, and that is tissue. Okay, tissue. So here I show you a picture of the monstrance. This is Lano, 700 AD, 1,300 years ago. That's what remains now. And when the scientists examined this, he said, this is heart tissue. This is a perfect cut through the heart. It shows the chamber called the ventricles of the heart, okay? And 700 AD, we didn't even have sharp enough tool to make a tiny, very thin cut, right? But for this to have occurred, it's a miracle, because no man could have created this as a fake, okay? But they looked at it 1,300 years later and said, oh, this is heart tissue, okay? And they did say it's not quite normal heart tissue, but it's definitely heart tissue. So I bring you to the devotion of Sacred Heart of Jesus, all right? So when I read this part, I'm like, oh my, Sacred Heart of Jesus kind of brings up a new meaning for me, okay? Because that's what showed up. And it wasn't just the Lano miracles that showed the human heart. All 5 miracles. Buenos Aires, Tixla, Mexico, Sokolka, Poland, Linica, Poland, all 5 showed heart tissue, human heart tissue, okay? Now, with the exception of Lano, Italy, the other specimens showed a damaged heart that has been beating violently, out of control, because of a humongous amount of hormones, catecholamines, racing through the body because of tremendous stress. So this corroborates the findings in the other test, right? Does that make sense? Remember the blood test specimen? Okay, so the heart specimen shows that the heart is heavily damaged, so much damage so that a lot of the experts were not able to definitively say, it's heart tissue. So that's when Dr. Zugabe came into the scene. Dr. Gomez looked up who's the heart expert, found Dr. Zugabe, and sent Dr. Zugabe. In fact, he came with a camera crew and said, Dr. Zugabe, can you please look at this slide and tell us what you see? And Dr. Zugabe, being the expert, said, sure, he's top of the line. I mean, he knows his stuff. So he looked at it, and he said, this is definitely heart tissue. Okay, I've seen this before. And he went on to describe what it is. He also said that there are white blood cells in the heart tissue. Okay? Quick biology lesson. White blood cells circulate in our bloodstreams. They are your immune system cell. They are there to prevent any attack. They're there to protect you. Once they circulate and they find something wrong, maybe you have an infection, the white blood cells will leave the blood and go to the area that's damaged. So in this case, the white blood cell circulates around the body, went to the heart because the heart was heavily damaged. The white blood cells will come to the heart and start to try to prevent the damage and then try to eat up the debris because it's no good anymore, right? That's exactly what Dr. Zugabe saw. He saw different types of white blood cells. The first wave of white blood cells that comes to kind of stop everything. And then the second wave of white blood cell came to start cleaning up the mess, okay? So what does that tell us? Number one, Jesus is alive in the Eucharistic miracle. The heart is alive because white blood cells can only live outside the body for minutes, an hour max. If the white blood cell is outside the body for longer, they will die and they will start to break apart and it won't be white blood cells anymore. So when Dr. Zugabe saw these white blood cells, the new and the cleaner upper, he said, oh my, when you took this specimen, the person was alive. Yeah. And then he said, he went through all this, and then he turned to Dr. Gomez and said, so tell me the story. And Dr. Gomez said, this has been sitting in distilled water for 3 years. And at that point, it's just, wow, So Dr. Zugabe, being the expert, was able to call heart tissue. And then from that point on, other scientists also say, yeah, he's correct. It is heart tissue. It's heavily damaged. It's hard to tell. It lost a lot of the features, but it is heart tissue. So it's confirmed by multiple scientists that we have living, dying, heavily damaged, tremendous stress. And Dr. Zugabe said this person might even suffer a heart attack because if you fell on your chest, the impact is going to be blunt trauma to the chest, and that could result in the heart attack, the myocardial infarction. Okay. So the heart was tremendously, basically dying, which is consistent with, remember, the blood finding. The lymphocytosis and the hypogammaglobulinemia exist because of tremendous stress. Okay, so there's confirmation of what's going on. Okay, very interesting. I got to throw this out. So the scientists, who looked at this, they had a conference. One scientist came up with a hypothesis for cause of death. Now, back in the old days, Roman, they crucify people because they say, well, they're going to die of suffocation. But that's not happening with Jesus, because the signs of suffocation is the person going to be in and out of it. They're not quite there. But in Jesus' case, he was very coherent. He spoke loudly and logically and clearly. So he was not being deprived of oxygen, okay? So what caused his death? Because he died pretty suddenly. Okay, well, one explanation would be, let's take a look at the heart here, quick anatomy lesson. So the human heart sits inside a sac, right? And the sac is very tough and very strong. So let's say that Jesus' heart was tearing. It was ripped. It was beating out of control to the point where it's so violent that the heart fiber was torn. If that is the case, then the bleeding from inside the heart would bleed into the space in the pericardial sac. That pericardial sac is not a lot of space, folks. You know, it doesn't take long before that sac starts to fill up with blood. And when that happens, the heart can no longer beat. The heart is like stuck, and that's death, okay? So that could be what happened because Jesus died pretty quickly, right? He died pretty suddenly because if his heart broke, and then my husband would say, oh, Jesus died of a broken heart. And it's true, he died of a broken heart for us, literally, right? So that would be his immediate death. Okay. And so let's continue the story. So as some of you know, when blood stops circulating, it's stagnant, it will separate, right? So the lighter stuff will go on top, the heavier stuff with gravity will settle to the bottom. So the lighter stuff are your plasma, that's the fluid component of blood, and the heavier stuff would be the red blood cells. Okay. So we know from the Bible that the Roman soldier came through, looked up, was going to break Jesus' leg, but saw that he was already dead, right? So what did he do? Thrust the spear into the side, and out came blood and water. Now, if you were back in that time, you look up and yeah, there's this red thing and there's this not so red thing coming down, so you may call it blood and water. We now can call it, could be red blood cells and plasma. So that actually makes sense too, doesn't it? Okay, so it's a hypothesis. There's no way we're going to prove that to be true, but it is consistent with what we're told in the Bible. Okay. All right. Science and faith. You know, I'm passionate about this topic because I am now seeing how science supports faith. Too often we hear people saying, well, you know, science negates faith. You know, you believe in science, you believe in faith. And then it's, no. Now we get to a point where we can actually use science to validate what we believe. And that's why I'm thrilled to share with you. You know, I can stand on the rooftop and just shout it out, right? Because this is what we need to understand. Our faith is now supported by many, many scientists all over the world, okay? And it gets better. Those who claim forgery, we can actually say, yeah, if it is a fake, the scientists would tell you it's a fake. But if it's for real, the scientists would tell you this is supernatural. So let me give you an example. So Tixla, Mexico, remember the religious sister was giving communion and she looked down and was like, oh, okay, there's something red here, right? So she took that to the priest, and the scientists examined it. So the bishop said, can you tell me if someone could have dropped blood on top and then call it a miracle? And the scientists said, okay, sure, we'll try to do our best. So with a very high, powerful microscope, what they did was they looked at Jesus, not top down, but on edge, on that very thin edge, right? So with a very high power magnification, what they saw was on top was dry, crusted blood, and deep to that was fresh blood. Fresh blood after 3 years. Oh my goodness. And it gets better. So let's say if this is the thickness on edge, right? This is the thickness because it's highly magnified. What they saw was a cone. A cone. So the bleeding came from one spot in the host. And it's as if that bleeding spot just started pumping blood out. So it started to go this way. It's a cone. And when it got to the surface, it went over, right? Whereas if you were to drop something on top, it will soak through evenly, right? It will soak through evenly. There's no way it will make the cone-shaped figure. So the scientists said, highly unlikely that someone dropped blood on top because of this cone shape. There was bleeding from one pinpoint spot, and that, as if it was the heart pumping blood, and blood bled. That's why there's still fresh blood deep. The top is dry, crusted blood. Wow. How can anyone forge that? Right? You can't. So it's supernatural. Yeah, so, you know, like I said, all of this is exciting. It's, actually, I'm going to skip that. So I'm trying to wrap up with, you know, I've been thinking about this for 6 months now, 7 months. And so I'm a little ahead of the game, whereas for all of you, I've asked you to come home, bring it, and think about this, because it's just so powerful. There's just so many different angles. So I'm going to share with you a few of my reflections. For me personally, I dropped to my knees at a good, merciful, generous, gracious God, because that's how I feel. You know, me, who should believe, am struggling with belief, and instead of punishing me for not believing, God provides evidence. Time and time again, he shows us how much he loves us by not punishing us, but trying to help us to understand. And that, to me, is a wow kind of reflection. You know, the tremendous love of God for us, okay? Another reflection, true God and true man. The DNA, the heart, the blood, all the scientists said this is human. Not an animal, not a fake, human. But the DNA, the fact that we cannot profile Jesus, that to me is divine, right? So Jesus is human and divine. And now I would say that's scientific proof. Everything about the miracles were human, but we couldn't profile the DNA. Okay? God does not change. All the miracles, time and time again, same results. No matter who examined Jesus, same outcome. God's constant. We change, but God is always the same. Okay. And this one I learned from Father Spitzer. Father Spitzer is a priest and also a biochemist, so he's also a science geek. And so he speaks my language, and I went to his talk, and oh, by the way, he talked about 3 of the miracles. So anyway, this I got from him. Father Spitzer said, time is a human construct, but for God, there's no concept of time. It's almost as if God can collapse everything and expands everything. So when I'm in awe of, 1,300 years, Lanciano, Italy, for God, it's like, it just happened. And it's even better, you know, according to Father Spitzer, you know, that Last Supper, when Jesus, with his disciple, said, "This is my body," it's as if at Mass, every single Mass, the priest in persona of Christ. But I would say, you know what? At that moment of consecration, Jesus is there. We would be the disciples. And Jesus held up and said, "This is my body." You know, we say, oh yeah, you know, the priest is in persona of Christ, and the consecration happens. But Father Spitzer said, every time we celebrate Mass, we go back to that Last Supper scene. Jesus is there, and he is saying, "This is my body." Because to Jesus, to God, there is no concept of time. That happened, what I thought, you know, over 2,000 years ago, according to God, happens every time we celebrate Mass at the time of consecration. I'm like, wow. So yeah, God is infinity, right? There's no beginning, there's no end. So there's really no concept of time. Everything is now and forever. I know that's a hard one to grasp, so you need a little time, just think about that. And so I leave you with, how would this impact your faith? I hope you got some interesting story, which is great. Like I said, I thank you for being here. But really, we're on the mission. We're on the road together. And we're going to walk toward Christ. And our loving God are saying, if you are having a hard time believing, believe. Here's the evidence. Our scientific community is finally caught up to it. And so I will say, ever since I read this book, when I come up to receive Jesus, when I go to adoration, I'm reminded of how much God loves us. And I'm just like, wow. And I would do whatever I can to tell as many people as I can, because we all need to feel that wow, and be moved and believe. Thank you. We have a little bit of time for questions. Yes, sir.
You said that Jesus has AB blood.
Could you?
You said that Jesus has AB blood,
Yes.
and then you said that blood typing didn't come up till 1930. So how can we say Jesus has AB blood?
Right, they
Yeah.
discovered.
But that still isn't answer the question. How do we know that Jesus had AB blood?
Okay, so they created the ABO blood system in the 1930s. But remember, Jesus is examined recently, right? In 2013, et cetera, right? So since 1930, we know to call this A or B or AB blood. It existed way back then, but we didn't know what to call it.
So you're basing that on these miracles then?
What do you mean basing on the miracle?
That saying it's AB blood from it coming up as AB and all of these miracles?
Yes. So the system that was created by the international community of scientists to say, okay, if you have this, you consider A blood. If you have this, you consider B blood. So using that system, we are saying Jesus has AB blood. And the other thing that's kind of interesting is, AB blood is the universal recipient. You have AB blood, you can take other people's blood without any problem, which is actually kind of interesting because Jesus receives everyone, right? So, yeah. Any other questions?
Is it possible that the trauma to the heart was due to Jesus having to take in oxygen, which means the lungs are working harder, and on the cross, it's very difficult to breathe. So is it possible that the heart's needing oxygen from the lungs, and of course the lungs are working overtime, and then of course the heart is taking that impact because it needs oxygen to make blood?
Well, because the histology, looking at the slides, shows so much white blood cells and so much damage in the heart, it's probably the heart that was damaged first. And when the heart stopped beating, everything kind of backflowed, right? So all the blood that was in the lungs didn't move anymore, became stagnant. So basically that developed into fluid in the lungs, if you will, which is why when Jesus was taken down from the cross and the sudarium was wrapped around his head, he had bodily fluid coming out of his mouth and his nose. Those fluid that built up in the lungs because it had nowhere to go came out. And so it's consistent with the findings of the sudarium. So the lungs are protected in the rib cage and they just inflate and deflate, but really it's the heart that keeps the blood flowing. So when the heart stops moving, we're in trouble. with the stories of the three falls and all the whipping and all the beating and the tremendous trauma and the hormones that's racing through his system, is probably going to result in the heart damage much more than lung damage.
In the left ventricle?
Yes. So it's just interesting because Dr. Zugabe, when he looked at the slide, said, this is the left ventricle, close to the valve. I mean, I'm getting a little technical, but he pinpointed exactly where the specimen was, which was taken from the miracles, right? So Jesus showed us not just the heart, is the left ventricle, and that's how come we know it was tearing. It's a matter of time. When the left ventricle is the ventricle that pumps blood through the whole body, very thick wall, very strong, and when that tears, you have extremely high pressure going into the pericardial space, and that quick death. Yeah, that's quick death, yes.
I read that there's a blood condition where, like Jesus bled through his pores during the, when he was at the Garden of the Olives and suffering. What's the name of that condition? I forgot. Where Jesus was sweated blood.
Yeah.
Before his passion, and then he was arrested.
Well, it's once again, that condition corresponds, I don't know the name off the top of my head, but that condition is usually tied with a very traumatic, life-threatening event. When we were under so much stress that we start to sweat blood. So you're right, it does exist, it's very rare, and we know from the Bible that it did happen, and that matches the whole picture, right, of the damage that we see in the heart and the abnormal blood. So it's all consistent. It all happened within a very short time frame. And prior to that, though, Jesus was healthy. So yeah, you're correct. It's corroborate that tremendous stress.
Thank you.
Yeah. There's another question over there. Okay.
Are there any current miracles being investigated now?
I'm sure there are, but our church, in her wisdom, want to investigate it, right? So all the miracles that happens has to be scientifically studied. Now that we know all of this, before any announcement will be made. So remember, these miracles happen, but it took years before the results were released, because sometimes they have to consult multiple labs. They don't just go to one lab. So let's say, the example, Argentina, right? 1992 was when the miracles happened. The results weren't released, announced until well into 2000 something, So many years will pass. So there are miracles occurring now, I'm sure, but the church is not gonna make an announcement until they for sure have credible supporting information. And it's for the right reason, right? Because we don't want to, make the wrong announcement. So yes, there are miracles happening. They're being investigated just, over time.
This evidence is really compelling. I'm wondering, why do not more people know about it? Are people trying to prevent others from learning about this?
I don't know the answer to that, but I can tell you how we got here was because of someone who heard the presentation said, hey, I know people in Seattle. My husband and I, we're willing to travel and share the message, but not everyone believe us. I can tell you, we've been to different places, and I would send emails out, say, hey, hello, pastor, father so-and-so, we're a deacon couple, we do this thing, and we have this webpage. On my own, zero. And I've sent out a lot of emails. But I guess, people are hesitant because they don't know us. So far, we've been to, I would say, 25 different groups. And all those groups was because of referral, word of mouth. Someone in the office heard it and took it back to their parish. And so I extend the same invitation to all of you. Help me, because on my own, my email goes unanswered. Okay. Oh well, they're not rejecting me, they're rejecting Jesus, right?
Thank you, Doctor, and congratulations. Welcome home. From the genetic point of view, the chromosomal X and Y, I understood that they had identified a chromosome X, but not a Y. Can you update if they have found something new or different, please?
Chromosome Y did exist. That's why they know Jewish male, right? Is that your question? Chromosome X. Okay, you mean the sex chromosome? Okay. Right. Okay. So a little bit of biology. XY chromosomes are male, right? The X chromosome is a lot bigger than the Y chromosomes. There are certain genes that exist there are many, many genes because X is a lot bigger. There are many genes that exist on the X chromosome but not on the Y. And they did look at the X chromosome to look for specific genetic information. So it was examined. I don't know what exactly what they looked for on the X chromosome, but it's in the book. Dr. Serafini will write up about it if the scientists did examine the X chromosome. Okay, yes.
Sort of in line with that, can you just talk about, was it really special that they could get information from the DNA, but very strange that they could not get a fingerprint? Or was it likely that the DNA was just damaged and they couldn't? Or was it very unique in both?
I don't think anyone really understands. Especially the Sokolka and the Lanika Poland, because it just happened and the scientists examined it within a matter of months. They believe they had some pretty good samples. Okay, of course, Lanciano and Tixla and Argentina is so long ago. 3 years is a long time. So yes, the DNA was not in great condition. But even with the good specimen, they couldn't profile. Now, God, okay, so this is also in the book. God knows so much of where we're going to go with this. So one of the scientists hypothesized, we do cloning now. And if you have the profile, you have the exact 3 trillion base pairs, there's going to be some mad scientist who's going to try to clone Jesus. You know what's going to happen, right? We have the ability to clone right now. We have cloned animals, and we can clone human. Has it happened? But that was one of the possibilities why God is like, we're not going there, I don't know. I don't know why the laboratory say that they could not profile the DNA of Jesus. Yeah. Yes. Oh, sorry.
Lonnie, first, thank you. Thank you for sharing your excitement and your joy for what we've learned. It is contagious and inspirational. One of the slides caught my eye, and I don't think you touched on this point, but it was when you were showing the analysis from the DNA fingerprinting.
Mm-hmm.
did I read correctly that they were able to determine that the specimen had a human mother, but they couldn't determine the father?
That was another possible explanation that we know from the Bible, right there, that Jesus has a biological mother, but the Holy Spirit is the other half. We don't know, but okay, let's put it this way. For us to profile the DNA, you pretty much have to have the whole thing, right? Because we're looking for a specific segment. And when you don't have those specific segments, you can't profile. So the segment that the scientists did see, they can say is human. And specifically, humans have a specific hemoglobin gene. So that's how we know it's human, that hemoglobin gene is there. But are there pieces that are missing? Possible. That could be a reason why they couldn't profile. Why are they missing? Could be because Mary and Holy Spirit, you know. So that's where I think about it, and I'm like, gosh, true God and true man, right? True God and true man. Yes.
It's like what is Mary's what is Mary's blood type?
Oh yeah, we don't know.
I mean, it goes it goes along that line, the lineage, the
right.
introspective.
Well, Mary would have to either be A or B, right? For Jesus to be A, B. She can't give Jesus both A and B.
Right.
Yeah, but I don't know if we have any.
But then she had the mother and father too.
Yes. So it'd be interesting to profile Mary. Yeah, profile Mary. And then compare it to Jesus and see, are they related? I know. All right, any other questions? Yes.
I was just wondering, when they send these samples to different labs and to people, especially to people who don't know what they're examining, what sort of precautions or procedures are taken with it to still reference it and make sure it's still safe and good, or is that kind of, is there, how does that work?
Yeah, I'm kind of hard of hearing you that. I'm very nervous, so can you just repeat the question?
Yeah, so when they send the samples to different labs, how does that work to keep it kind of safe and still referenced?
The procedure is handled by Dr. Gomez for the Tixla and the Argentina miracles. And Dr. Gomez, after the first experience with Argentina, he made it a point for him to handle the process and videotape how it's sealed and videotape how it got to the lab and to be open to ensure the chain of custody is not broken. So the latter miracles, stuff in the 2000 and beyond, they are very definite, very careful to document chain of custody. When the Poland miracles, those scientists came to the site to take, and same thing, they recorded who handled it and how it is packaged. So it's very well documented. Yeah, thank you. Where they are, they're where they are. So the miracles in Lanciano, Italy is in Lanciano, Italy. Yeah, so it's still there. These scientists took minimal samples because they knew they take it, it's all gone, right? I mean, you can't bring it back. They really careful with how how much. And so the bulk of it is still in the monstrance, in air, so all over the world. You know, it's my bucket list. I'm going to go and check off these things. Yeah, it's very interesting. If you read the story, Dr. Franco Serafini talks about how each experience was so different from each other. Yeah. Yes. Another question? I know it's getting late, so if you need to leave, feel free. I'll hang out to answer as many questions as you have.
First of all, I'm so thankful that you're doing this. I feel like hugging you.
I'll come and hug you. Oh, thank you. Oh.
Second is Blessed Carlo Acutis, whom we've adopted as a parish saint.
Thank you.
He's prepared a website of about 100 Eucharistic miracles. At St. Mary's in Anacortes, they had an exhibition, maybe in the late 1980s, when they had a conference, Word on Fire. And at every World Youth Day, they have an exhibition for the pilgrims of all the Eucharistic miracles. But what I appreciate the most tonight is, by Dr. Lani, whether the consecrated host was found on the floor, in the ciborium, on the sink, whether it was sent to the labs. She always said Jesus was sent to the labs. Jesus was on the floor. It was not a specimen. It was not a host or a consecrated host, but I'm so happy that you called him Jesus. Thank you.
Thank you.
And we're actually going to have, on the weekend before our parish mission, February 24th and 25th, we're going to have an exhibit that shows all of the Eucharistic miracles on Carlos Acutis' website. So we'll be getting that information out, and then our parish mission, Father Ed's going to lead, and it's going to be on the book that was given out at Christmas by Bishop Barron, This Is My Body. So Lonnie was a perfect intro into that, and that'll end up being the last week of February, so we're going to be getting that information out. And this is the year of the Eucharistic Revival, which, if you've been watching the bulletin and the update, we have a link that you can go to, because it was the Pew Research that moved the bishops of the United States to start this Eucharistic revival, because they realized how important and necessary it is for Catholics to understand the real presence of Christ in the Eucharist. So I really encourage you. We're going to have a revival night once a month, and we're going to show DVDs that have come out of the revival, the national revival, and a number of people are going to the conference in July. So if this has whetted your interest, be sure to continue with us, because we're going to do the exhibit, and Father's going to continue with the parish mission. But it's 9 o'clock, so our facilities crew would like us to head home. So thank you all for coming. This was live streamed, so share it, And thank you, Lani.
Thank you. We're great. Oh, okay, awesome. All right, praise God. Okay.
Thank you, doctor. Thank you very much.
Well, thank you.
You're so cute.
Sure. Thank you.
That was wonderful. Absolutely wonderful.